Hello Mr. Manny in class. Today I'm going to be presenting on the ethical issues of using CRISPR to modify livestock. What is CRISPR and the Cas9 protein? In short, and to summarize what I'm about to say, using CRISPR and the protein Cas9, genes can be modified or taken away, and using polymerase, even have new strands added that can dramatically change the genotype and the phenotype of an organism. Before we get into the ethical issue of CRISPR, we need to first understand what it stands for. So, to begin with, the S standing for short and the R standing for repeats give us a clue. There are short repeats. These are found every 40 pairs of nucleotides, the nucleotides being the letters A, T, C, and G. The P stands for palindromic, meaning the letters can be read either way. The I, standing for interspaced, means that between these repeats there are strands of DNA called spacer DNA. The C and the R, clustered and regular, tell us DNA is found in clusters and that we find spacer DNA strands at regular intervals. Next, I'm going to explain some CRISPR background and show you a GIF of the Cas9 protein making a cut in an RNA strand. Cas9 naturally occurs in the body as a protein. It is used to help fight off viruses. The protein has an important ability that allows it to read DNA and identify repeats or unwanted strands of DNA that could contain a virus. We all know viruses rely on their genetic code to churn out more copies of themselves. The more cuts and misinformation the virus has, the less likely it will be able to copy itself effectively. In the bottom right of the screen, you're about to see the Cas9 protein performing a cut on a strand of DNA. As you can see, the Cas9 protein has wrapped itself around the RNA, creating a cutting site. At 29 seconds, you can see the Cas9 protein has successfully cleaved away the DNA, making it impossible for a virus to read. So that's what CRISPR and the Cas9 protein does naturally. But that's not it. In 2012, a group of scientists from America and France discovered a use of CRISPR in gene editing. It involves reprogramming the Cas9 protein to cut out specific sequences. So here enters our dilemma. In this next section, I'm going to talk about the morality of gene editing. I'll begin with a story. It's made up, but this could be plausible. To produce milk, cows put aside lots of resources in their body. This is why we need to feed them so much grass. Let's say in this hypothetical world, cows are eating too much grass. There's not enough grass and there's too many cows. We could use CRISPR to slow down a cow's metabolism, meaning they would require less grass to eat. And now this is great. The cows are producing the same amount of milk with less grass. And our pool of cows, the small sample, works so well. The idea is adopted worldwide, and cows worldwide have a slower metabolism while still producing the same amount of milk. The normal lifespan of a cow is 48 years, but let's say in this scenario these cows are starting to die at the age of 28. In this hypothetical world, it turns out that slowing down the cow's metabolism puts too much pressure on their stomach and leads to a premature death. In this scenario, we've killed millions of cows across the world and experienced billions of dollars in losses. This is an example of an issue that could arise. It's plausible we could see a world in which CRISPR begs us to ask the question, is anything natural anymore, and could this research be transferred over to humans? We've seen this in pop culture. Movies such as Gattaca express to us the problems with CRISPR and gene editing in humans, presenting us with a dystopian future. While CRISPR-Cas9 gene editing could lead to extraordinary advances in medicine and science, it's important to understand, quote, "The road to hell is paved with good intentions." CRISPR isn't all bad, however, and there are many positives that could come out of this field, including what I'm about to talk about now. Understanding how CRISPR works in animals, it'd be much easier to help translate over to humans. Using CRISPR on humans could help us cure and understand disease, as well as effectively giving us superpowers. In a report by the New England Journal of Medicine, working with many students and professors, there was an attempt at using a programmed Cas9 protein to effectively turn off a carrier protein that allowed the HIV virus to infiltrate the body. The test proved around 5% accurate, And while a long way off the 95 to 99% Goldilocks zone, it was a step in the right direction in order to help hundreds of thousands across the globe. And in another report, as of early March 2020, a detailed method of combining gene therapy, which is the replacement of injured or defective cells with new cells, and CRISPR-Cas9, could lead to new advances in not only curing disease, but also repairing damages said disease might have caused. What's amazing about this research is that it is quite transferable over to other living organisms. The same knowledge used to help cure humans could also benefit animals. Using CRISPR in livestock could also present solutions to world hunger, or maybe just a cheaper carton of milk. Going back to cows, this time with a less gruesome scenario, the FABP4 gene in cows is responsible for producing milk. We could use programmed Cas9 proteins in cows to edit the yield of milk, proteins, and even some of the texture and taste of milk entirely. Increasing the yield of milk per cow would also result in less cows across the globe, and seeing as livestock produce 14.5% of the annual greenhouse emissions, this would cut down on climate change dramatically. Excusing the pun that obviously had to be made, the stakeholders in the new CRISPR livestock would include farmers, who have to make money off milking, shearing, eggs, and fur. These would be the people most directly impacted. CRISPR making life for farmers more efficient would mean farmers need to put less work in, but would still reap the same rewards. Activists for animal rights would also be affected. There would definitely be the possibility for this technology to be misused, and its regulation would be very important. Breeders of livestock would also be negatively impacted. If it were possible for farmers to create their own cows, prize breeding would become irrelevant and would destroy the market. And finally, but most importantly, the animals affected by CRISPR should continue to at least have a life in no more pain than lives they currently experience today. Ultimately, CRISPR is a tool that, while still in its infancy, promises a pool of new advances in science. And while CRISPR will provide many extraordinary applications, it is important that we stay within the ethical boundaries of this new technology. Thank you.